questions about viruses or for that matter, the classification that we just finished up? Thank you for coming, Latoya.
I looked my first.
Ooh. Go. Yeah.
A retrovirus.
Say that.
What is a retrovirus?
A retrovirus specifically has RNA instead of DNA.
Okay.
Period. That's a retrovirus. And the catch of it is that the RNA then has to produce DNA, which is basically forbidden, except that they do it. But because it's forbidden, the base sequence is all screwed up basically, and because of being screwed up, it just causes terrible diseases. But the main thing is RNA will produce DNA from the retrovirus.
Wouldn't it have the U and
Well, the RNA will, but when it makes DNA, it's going to have anything else? Okay, well, let's keep going then, because actually we're already through most of this for the viruses. Let me just remind you, though, the whole point that they might cause cancer is because that they're going to insert their DNA or change the host DNA. And in changing the host DNA, who knows what you're going to get? It changes the properties. Maybe. Now, maybe nothing. But change the properties possibly. Changing the properties might lead to cancer. Or maybe nothing. Okay. So the most common product of these oncogenes, remember, all nucleated cells have oncogenes. We all have genes that can cause cancer. Now, clearly, most of us will never get cancer. So that means that our genes are normally inactive. But something activates them, maybe. Something activates them that causes cancer. So, what I have here, the most common product of oncogenes are kinases. Notice a mistake. And I can't blame anybody else. This mistake is mine. I say that kinases transfer ATP. Well, as you know, that's not quite true.
Phosphates.
Yes, indeed. They transfer phosphates. And granted, yeah, usually they'll transfer phosphates from ATP, so I'm trying to justify myself, but basically I made a mistake. So please change that in your notes. Kinases transfer phosphates. What you learned from glycolysis is true. And as happened in glycolysis, quite often transferring the phosphate can play an activation role. Now, it doesn't always. You also saw that with glycolysis, but it certainly can activate. And that's probably what happens here. By transferring the phosphate, you might activate the oncogene, causing cancer, and off it goes. So again, make the change. Obviously, kinases transfer phosphates, probably from ATP, but not required. So that might play the role in activating the oncogenes. Viruses that activate oncogenes are called oncoviruses. And usually, they're lysogenic viruses, usually. Yeah. Now, converting normal cells, the oncogenes are going to do that. They're going to convert normal cells into cancer cells. And this is called transformation. Now remember, how exactly that occurs is not known. How are normal cells transformed into cancer cells? But one thing is clear. Contact inhibition is inactivated. Now this actually is one of the very few contributions I myself made to science. I was working on my degree, which had nothing to do with cancer. I ran into, well, contact inhibition is why normal cells do not become cancer, normal cells. Because when they're growing, they get close to each other. When they get close, they stop growing. That's the contact inhibition. If they get too close, they stop growing. Nobody's ever known how it worked. And if you don't know how it works, you don't know how to fix it if it goes bad. So working on my degree, I basically, again, this is off the record here, but it deals with this. I found out that cells have on their cell membranes these molecules called sialic acid, again, off the record here. And despite the name, they're actually carbohydrates. Well, the relevant thing of this is that sialic acid has a negative charge. So essentially, what was going on, that all these cells had negative charges. And when they would get close to each other, essentially they would undergo electrostatic repulsion. All the negative charges would repel each other. You got lucky. All the negative charges would repel each other, and that would be the contact inhibition. Actually, fairly simple, obviously, but nobody had seen it. You might wonder, this is one of the things I had to look at, you might wonder, cell membranes are negatively charged. Why wouldn't cell membranes just simply repel? Actually, not strong enough. All cells have negatively charged cell membranes. But the phosphates in them, they're buried in the membrane, et cetera, is not a very strong charge. So cell membranes did not work to repel. So anyhow, all these negative charges from the sialic acid help to repel, basically the cause of contact inhibition. Well, clearly, cancer cells don't seem to have contact inhibition. They grow like mad. Got a car one? They grow like mad. So something deactivates the contact inhibition. Again, nobody knew what caused it, so they weren't able to figure out what happens when it goes wrong. Well, now with my sialic acid thing, there's an obvious implication that maybe with cancer cells, the sialic acid is removed. Now, how it's removed, I don't know. But I was working in the hospital, so I went down and got some tumor cells. You can do that if you're in a hospital. So I got 3 different tumors. There's actually about 100 tumors, different kinds of tumors. I got 3. And I checked out to see if sialic acid was on the membranes, because all cells have sialic acid. And it turned out, no, they did not have sialic acid. So I was really excited and thinking, oh boy, Nobel Prize for me. And so I published my results, sent them off. But then I was starting to think, well, if I'm going to get a Nobel Prize, I should probably get my degree first, so it sounds better when they give it to me. So I finished off my degree and stuff, but meanwhile, some people read the paper, which is what happens. And some guys who actually knew what they were doing, meant they actually knew something about cancer, checked it out, which is what they're supposed to do. And essentially tested a lot of other tumors. Like I said, there's actually about 100 different kinds of cancers. So they did a lot more than 3, like what I had done. Turned out I had gotten lucky. That the 3 I had tested, in fact, had no sialic acid. But a lot of the other ones that these guys tested did have sialic acid. In other words, it was wrong. That's why I'm here, and that's why I have no Nobel Prize. So anyhow, the contact inhibition thing is still true, but we still don't know why the cancer cells don't have it. If somehow we could give the contact inhibition back to the cancer, that would solve cancer. It would stop growing. Anyhow, stay tuned. I'm sure somebody's going to figure it out, but we haven't yet. But for whatever reason, we're back on the record, by the way. For whatever reason, the cancer cells do not have contact inhibition, so they keep growing.
Is there a way to get?
Because apparently, whatever gets rid of that stuff to begin with, does it also? No. I did talk to you last week, didn't I, about why cancer kills? Mm-hmm. I did that. Yeah. I thought I did, but I wasn't sure. Okay, so there's actually 4 different possible mechanisms to activate these genes. Remember, activating the genes is equivalent to causing cancer. So one, basically, is bad luck. mutation. Now that can happen to anybody, that there's a mistake. By the way, our genetic machinery, replication, sequence changes. If you change the DNA makeup, you change the DNA product, meaning different proteins, that might cause cancer. Again, it might cause nothing. I keep saying that. It's just random good luck or bad luck. Sometimes you get a bad result, sometimes you get no result. Anyhow, mutation. Second, transduction. We talked about that the other day. That is when the lysogenic virus infects the cell, stays dormant for a while, and then when it becomes active, the host DNA becomes deactivated. Only the viral DNA replicates, but sometimes a piece of the host DNA is carried along. It's not supposed to be. But again, as I've said to you before, it's not a perfect world. So sometimes a piece of the host DNA will be included when the viral DNA replicates, which means you're taking along some extra DNA. So that can cause cancer because the extra DNA, when it inserts into a new cell, well, you're changing the DNA again. So transduction can cause cancer, maybe. This one's kind of an interesting one, translocation. Okay, this means that some genes, not just oncogenes, genes in general, can move from one spot in the chromosome to another. Contrary to what you've probably always been told and thought, genes do not always stay in one place. They can move in the chromosome. And once again, if you're moving the genes, you're changing the genetic makeup. And once again, who knows? So changing the genes, having the genes move around through nobody's fault, might lead to a cancer thing. What's interesting about this is the story of the person who discovered this. Her name was Barbara McClintock. And contrary to what you probably think about scientists and stuff, that they're just trying to figure out what's right and what's wrong. Nah, they're jackasses in science just like everywhere else. And there's politics in science just like everywhere else. So this woman, Barbara McClintock, and this is now, 50 years ago or so, 60 maybe, but 50 years or so, she came out with this theory that the genes can move around. Well, many of the male scientists simply did not believe that a female scientist could come up with such a proposal. They didn't believe it, they belittled it, and they called her theory jumping genes, instead of jumping beans, jumping genes, and basically made fun of her. She's a woman, how could she possibly come up with something? Well, fortunately, not only was she proven right eventually, she was proven correct, but she lived to be proven correct. And she got the Nobel Prize. Probably mine. But she got the Nobel Prize for it. So she did, she was a very nice lady and all that. She did live to be proven right, to have these people laughing at her, proven wrong, and all the rest of it. She just died a few years ago, Again, this whole thing occurred 50 years ago, so she did well, At any rate, he came up with this, so there is politics and science just like everywhere else. But anyhow, the genes do move around, and I don't mean just oncogenes, any genes. They all move around. Not a lot, but they're not stationary either. Okay, finally here, gene amplification. As you're doing replication, you're making more and more genes. Sometimes the genes become active, and they might produce lots of product. And lots of product, sometimes you reach a threshold level. You have too much product. That might trigger cancer. Basically, gene amplification. You're making too much product, more than you need. We talked earlier this quarter about feedback inhibition. If you start making too much excess product, things might shut down. Yeah, they might. But they might do this. The too much product might cause cancer. changing the results again. So these are just possibilities. But of the 4 possibilities, 3 of them involve viruses. Only mutation does not involve virus.
breast cancer, someone
I'll present you before we move on is I want to go back to the thing with the 5 phases of viruses, infection, and so on. So attachment, penetration, biosynthesis, maturation. That's 4. Maturation. The 5th one is release. So the 4th one is maturation. That's when you've got the pieces, the viral DNA, the viral proteins, and now they're going to be assembled into new viruses. So that whole period is called the eclipse period. The eclipse period is when you've got the pieces that have not yet been assembled. So let me remind you that your virus has simply a protein coat . And of course, inside is the DNA. Okay, the protein, excuse me, the protein coat thing is not as easy as it looks. Because in reality, going back to your viral DNA now, it's undergoing transcription, undergoing translation. When the protein comes out, the viral protein, what you're really getting is that. You're getting a single protein. Excuse me. You're getting a viral protein, and the viral protein needs to be chopped up into pieces. By proteases. These are produced by the infected cell. So proteases will come along, chop up this big protein into smaller pieces, and the smaller pieces will be assembled into forming the protein coat.
So the protease is the lines that you see.
The proteases are the enzymes.
Okay.
Because ASE, that's enzyme.
Okay.
They will chop up the protein into
Okay.
smaller pieces.
Got it.
Into smaller pieces. Now, I may be off a little bit on my dates, but about 10 years ago now, the first bona fide treatments for HIV came out. HIV's been around since 1981, or AIDS has been around since 1981. The first published cases came out in San Francisco and LA. Of course, both of those cities have large gay populations. And obviously, since then, it's just exploded everywhere. But anyhow, so there have been other treatments not effective, and essentially, once you had HIV, death sentence. You were just going to die. Well, then about 10 years ago, they came out with this cocktail. They called it a cocktail. Protease inhibitor cocktail. They called it a cocktail because at the time, these people had to take 30 pills a day on a set schedule. It was really grueling. It worked, but it was very, very grueling. But that was the cocktail. And what this cocktail did, it contained protease inhibitors. And these inhibitors inhibited the proteases from cutting up the protein. To rephrase that, by preventing the protein from being chopped up, it was preventing the pieces being formed to make the protein coat. In other words, with the protease inhibitors, the virus was unable to make the protein coat, therefore was unable to make new viruses. So the downside is you had to be infected for this thing to work. It's not a vaccine. It does not prevent HIV. But once you got it, it prevents the spread of HIV. As you know, with the lytic cycle, lysogenic cycle, you're going to be making new viruses, the cell ruptures, the viruses go out, infect new cells. Well, with these inhibitors, with this cocktail, the virus was unable to make new viruses. It was making the new DNA, it was making the protein, but not able to chop up the protein.
But, okay, so in HIV though, don't your cells reproduce like too much?